So good evening, ladies and gentlemen. I hope you can hear me well, and also good evening to those who are joining us online. We are a slightly depleted audience this evening. Mike Martin, who is the coordinator of these talks, he unfortunately has got some sort of flu, but it would appear that much of the village is also suffering from variants of cold and flu, and so people are being very, very cautious this evening. However, I'm delighted to say that Dr. Andy Taylor has made it over from Aberdeen. Thank you for coming, Andy. Andy is from the Hutton Institute, as you'll have probably read in the and he's a colleague, of course, of Colin Campbell. You'll remember that Colin Campbell gave a talk two years ago all about the Hutton Institute, which was absolutely fascinating. Colin happens to live in the village, but apparently is on holiday, so he's not here tonight. So I'm fascinated by the topic that Andy's going to talk about. It's all about Munros. For those people who are Munros, relax. Yeah, I don't think he's going to criticize Munro baggers or anything like that. He's going to talk about something completely different. So Andy, thank you so much for coming. We really appreciate your effort.
Super. Many thanks. So hopefully this will work. Can't actually see the key. Okay, so thank you very much for the invitation and thank you for the introduction. What I'm going to talk about tonight is actually very fitting to be here because much of the work which I'll show you tonight has actually been done in this area, and it was done in conjunction with Plantlife. So we've been working with the charity over the last couple of years, and myself and Andrea Britton, also from the James Hutton Institute, have come up with some crazy ideas, and they have been very kind to work with us in following or helping us carry them out. So what I'm going to talk about is really what's going on under your feet up in the hills, and we're looking at soil biodiversity and primarily looking at fungi tonight. If you have any other questions about any of the other diversity, then I'm very happy to answer those. So just really as a reminder about soil biodiversity, sadly, much of it is hidden. You can't see it unless you make an effort to actually blow it up to the size where you can see it. But just to give you a reminder of how common it is and how diverse it is, soil probably contains about a third of all the biodiversity on the planet. I think that's a conservative estimate, really. And there can be tens of thousands of species in a gram of soil. That's about a teaspoonful. Okay, so you're talking incredible diversity in very small amounts of soil. And the biggest challenge we have in looking at it, measuring it, quantifying it, is really because it's hidden. It's hidden in the soil, and much of the diversity is actually microscopic. So even if you dug things up, you still wouldn't see very much because it's microscopic. And like I said, I'm going to focus on fungi tonight. I am a mycologist, somebody who plays with fungi, and that's what I've been doing for the last too long. So the fruit bodies that you see, the mushrooms that you actually see, are really just the tip of the iceberg. That's what the fungi produce. That's how they produce the spores. That's their reproductive structures. It's like having apples on the tree. That's where the reproductive parts of the organisms are produced. And fungi carry out all sorts of functions in the environment. I'll show you some of them tonight. Some of them decompose organic material. Others are parasites. And many of them, which I'll show you, are in fact what are called symbionts. They live in association with other organisms. They're really essential in the environment. Fungi are really the recyclers of the planet for all sorts of reasons. And they're very, very important in nutrient cycles, including carbon, phosphorus, and nitrogen. And without their activities, none of the plants which we see around us would be able to grow. And this particularly applies to the fungi which grow on the roots of plants, and there are many of them. Without the fungi associated with their roots, they would not be able to grow. And just so that you know, the lichens, which are a big component of montane biodiversity, they're in fact fungi, but they're fungi growing in association with algae. So the algae can photosynthesize, produce sugars, and the fungi can then live on those sugars. So that's a wee bit about the background of the fungi. What do we know, or what did we know before we did our study? And this shows the Cairngorm National Park, and that's all the records of fungi from 1850 up until this year. There's quite a lot of records, about 123,000 records of fungi. There's 3,700 species recorded. The great majority, or maybe not the great majority, but a lot of them are lichens of the records. Lots of records from forests and very few records from the mountain summits. If you look at that map, you can see the A9 effectively, and you can see the Spey Valley. So people get out their cars, do some recording, and off they go again. And that certainly applies, to mycologists. Okay, so lots of records, but very, very little information about fungi up in the mountains. Now, what are we actually looking at when we're up there? Because we think of them as alpine habitats, but there's not just one alpine habitat. Okay, when you're up there, you can see mosaics of vegetation, and some of them are very distinct. You've got large areas at the top right there, which is a grassland. You've got habitats which are dominated by mosses in the middle there, and then you've got habitats which are dominated by ericaceous plants, so the heathlands. Very, very distinct habitats, all dominated by different plant forms. And one of the habitats we were particularly interested in was down here on the bottom right, and that's the late snowbed. It's one of the habitats which is unfortunately under threat, and probably within the next 50 odd years, we may lose most of our late-lying snowbeds. And we were particularly interested to see what biodiversity may be in those habitats. So a whole range of mosaics up there. Oops. And one of our first studies, which we did 3 or 4 years ago, was on top of Ben Ann. That picture looks very odd. That's actually the top of Ben Ann. And the question which we asked was, is the above ground heterogeneity, which you see in the plant communities, is that reflected below ground in the biodiversity in the soil and also the functions of the organisms in the soil? Okay, so that was our starting point for looking below ground up there. So what we did, it was a bit of a crazy study. We took the MVC classifications, the National Vegetation Classification, and plotted out 99 plots up on Ben Ann. And we covered the 7 most common vegetation types. And then in 2018, 4 of us did 24,000 meters of ascent to set up these 99 plots. I wanted to do a few more so that it would be 3 times Everest, but they said no. But it was a huge effort. And what we did was we had these geoposition points in each of our vegetation types, and we did all sorts of things. I don't think anyone has ever buried 600 tea bags on Benan before, but it's a global study, and Lipton's must have made an enormous amount of money out of this, because there are two particular types of tea which are planted, or not planted, they're buried in the soil for 3 or 4 months, and you weigh them beforehand, you weigh them afterwards, and it gives you a measure of the decomposition that's going on in the soil. We also planted 600 lollipop sticks and did decomposition studies on them. So we had a whole range of things. We even had little buttons that measure temperature, and so we could work out how the temperature fluctuated, and in particular, how long the snow lay for in these systems. So we did a whole range of things, and that's a fascinating study in itself, but it's not the one I'm going to focus on tonight, but I'll just give you the summary of what we found. So those 99 plots, 7 vegetation types, and we found just short of 3,500 fungi species. Okay, we found all sorts of other things, but just to give you an idea of the number of the fungi, so 3,400 odd fungi species in those 99 plots. Okay. What was very strong in this data was that each vegetation type had a distinct fungal community. So what was going on below ground was really reflected in above ground. Okay, so different vegetation types had very strongly associated distinct communities. So that was one Munro, Benan. What about the other 281? Would we have the same pattern? Well, what we thought was we would scale up to the Cairngorms and take advantage of the numbers of people who are going up into the mountains. And we wanted to really take a pilot study to see whether or not we could work with walkers and charities to see if we could do what's called a citizen science project, where people engage with people like myself to look at sampling or collecting data from the environment. So we worked with Plantlife. They have a project going on in the Cairngorm National Park, and the object was that we would sample all 58 Munros in the park. So that's like about 20% of the total. So that was our objective, was to gain samples from all 58 Munros. So what we asked volunteers to do was to go up, and we sent them survey packs with instructions, maps, sample kits, and we asked them to climb their Munro and to collect soil from 3 predefined points that we defined. And we also gave them 1 extra tube for a lucky dip, so they could go out wherever they want, collect the soil, and we would tell them what was in it. So they then returned the samples with recording forms by post, and then they uploaded the photographs which we needed to be able to identify where their lucky dip was from. And I have to say, it worked incredibly well. We ended up, there were lots of people volunteering, so much so that all the Munros were adopted within about 2 weeks of this going online. We ended up with 73 volunteers. We've worked out various figures in relation to what they did. So it was about 477 hours of volunteering. We ended up with 55 out of 58 Munros, which I think is a fantastic result. We had 219 samples collected. And if you can work out the numbers of miles which they hiked, it's 890 miles, so that's Aberdeen to London and back. The amount of meters, which is crazy, is over 57,000 meters climbed. That's 6.5 times Everest, or halfway to space, apparently. So this is a great one. That was the number of calories consumed, which is about 1,100 bananas. Okay, so that just gives you a sort of summary of the statistics in relation to what these people did, which was fantastic. People seem to enjoy doing this. We got some very good feedback, and people were very happy to have an excuse to go up to their favorite hill and to collect samples for us and then send them back. So that was great. So what happened to all those samples? We checked all the photographs, all the altitudes, and we calculated the elevation and the aspect. We collected all the photographs and we identified all the vegetation types which had been sampled. And then the soil samples were processed in the lab. And I'll go through some of that process with you. So this is the cycle of sample preparation. Starting from top left there, the samples arrived by post, and we had a whole rota of people who were available to process the samples as soon as they came in. They were immediately sorted by hand, and any larger stones were removed, and then the samples were stuck in the -20 freezer until we had a batch of them. And then when we got maybe 40 or so, we started doing this whole process. Now going left from the top left, sorry, going right from top left, the first thing we did was to grind them up because we need them very small particles. And we did that with what's called a cryo mill. And it's a bit like a coffee grinder, except you put your sample in liquid nitrogen, and that's -196 degrees centigrade. So we need it cold to preserve the DNA because we're characterizing all the communities, all the biodiversity based on DNA. And although it's relatively stable, when you're grinding it, it does heat up and it can fragment. So you want to keep it as cold as possible. So we create a powder with our glorified coffee mill, and then we add some reagents and we extract the DNA. And that's basically what's happening down here on the bottom. The picture here is in fact a gel of DNA that we've extracted from all the samples. And each one of those little white bars is in fact the DNA from the samples. And that's what we signed off to get sequenced. And I'll explain what I mean by sequencing. Okay, so all the samples, all 219 samples went through this process. We extracted the DNA, but we also kept some of the sample to determine some of the soil characteristics. For example, we measured the carbon in the soil, we measured the nitrogen, and we determined the pH, the acidity of each of the samples. So it gives us some environmental data in which to look at what factors are affecting the fungi. Now, I wanted to show you what technique we use for characterizing the fungi, because you might have heard of DNA barcoding or community analyses using DNA, but I wanted to give you a simplified version to give you some more understanding. So what we do is we have a barcoding approach, and I'll explain it because this is based upon using barcodes like you have at the supermarkets. Okay, and each single fungus that we know, you can extract the DNA, and you then generate a little piece of that DNA which is unique to that particular species, and that can act as a barcode. Okay, when you go to the supermarket, all your messages or your groceries, they're all barcoded, and that barcode is unique. It's exactly the same principle. The DNA we extract, and there's a tiny little portion of it which is unique to that species, and that's what we use as a barcode for every single species. Okay, so we're going from one known species to a barcode that represents that species. It's easy if you've only got one species and if you've got one big fruit body. But what happens when you go into the soil? You extract all the DNA from that soil and you end up with that. Okay, loads of barcodes, all mixed up, all representing different organisms. And somehow we need to read every single one of those. You know, the cashier scans your barcode. Well, that's basically what we do, but we do it millions of times. Okay, so we end up with millions of these barcodes, and every single one which we generate can be a separate species. Okay, so we generate, or we read millions of these barcodes that we've extracted from the soil, and every single one can be a separate species. So you need some way of identifying what that barcode means. Fortunately, there are reference databases which contain barcodes which are known. Okay, it's the same as your cashier. You scan the barcode and that immediately links it back to the database saying what that barcode means. It's exactly the same as what we do here. So we have our barcode, we run it through the database, and if you're really lucky, you get a name. And I'll show you how lucky you need to be, because unfortunately, there are so many fungi, and we know so little about them that the databases are very poorly populated with reference material. Okay, so you'll see how lucky we need to be. So that's what we do. So that's what we've done with all the DNA that we got from our 219 samples. And I'll just give you some data on how lucky we were. That's the number of barcodes that were generated, over 4 million from those samples. Okay, you might think that's a lot, and it is a lot. Considering 20 years ago, it cost about £5 to produce one barcode. All right, we now produce them in the millions. It costs quite a lot, but the cost of a single barcode has gone down to something like 0.00001p. Right, that's how we can generate so many of these guys. Okay, that's the number of species that we found, 2,748 in that one study from the 55 Munros in the Cairngorm National Park. Okay, and this is how it breaks down. Of these, I use the term taxa there because the numbers that we could actually distinguish and put a name on was only 268. Okay, so 10% roughly of all those individual entities that we could distinguish, we could only put a name on about 10%. What I find amazing is that the number or the percentage that we could only say it's a fungus is actually higher than that. Well, a little bit higher maybe. Okay, so it's approximately the same. So all we can say is that that barcode is a fungus. That's all we can say about it. We don't know anything else about it. I find that extraordinary. It just shows you how little we know. So all the other 2,100 odd, we could place them in a genus or a family, but that is what we could only say was a fungus. Incredible unknown diversity. So what did we find? Okay, this is a good bit. So that's the number of taxa or species. Lots of different functional groups, and a functional group, by that I mean fungi which do different things. Okay, and the commonest group are the group that break down dead organic matter in the soil. They're called saprotrophs. Okay, fungi are just like us. We need carbon, we need sugars. Okay, the fungi are not plants, they have their own kingdom. Okay, fungi cannot produce their own sugars, just the same as us. Fungi, okay, so they need external sources of sugars, and these guys get their sugars from dead organic material. And you will have seen loads of these. Okay, these are the guys that are nature's recyclers. Without these, we wouldn't exist, because all the carbon, all the nutrients would be locked up. So So these guys break down dead organic material, whether it's plant material, fungal material, animal material. These are the guys that recycle it. Now, a lot of them produce fruit bodies, but the major proportion of them do not produce fruit bodies. They just exist as mycelium. That's the fine hyphae that these guys produce in the soil. These fine threads here are the individual hyphae that spread through the soil and break down organic material. Some of them, like this guy here, this is the first time this has been recorded in the UK. This is one of the fungi that we found, and you can guess where it was described from. Antarctica. That gives you an idea of the conditions that we have on the Munros in Scotland. And there's quite a few fungi described from Antarctica that we have found here, and they're all called Antarctica. So it gives you an idea of where they're actually from. Okay, so one of these guys. This is one of the fungi that only grows below ground, and you never see evidence of it above ground. But it produces spores, and that's how it gets around, but you'll never see evidence of it because it just exists as the mycelium. One thing I think it's important to realize, fungi get a lot of bad press sometimes. We hear about pathogens taking out larch trees, doing all sorts of damage, the ash dieback fungus. Okay. But in fact, pathogens and parasites are actually very common in natural systems, and they're part of the natural system. And we found lots of pathogens and parasites of animals, of plants, and also parasites of fungi. And they're very common. And this is one that grows on a moss. This is a very nice picture from Dave Genny from Nature Scotland. It's associated with mosses, but my favorite is this guy. The English name is called the Strangler because it's actually a fungal parasite, but what it does is its host, if you could call it a host, starts to produce a fruit body. It produces a stem, and then this guy takes over and it grows out from the stem of the other fungus. It's an extraordinary thing. And it's got a great name of Amanita contortipes. And there was one single record of this from Rothymarcus in 1957. And it was as a fruit body. That was the only record for this before we started looking below ground up in our mountains. Because we have found this. Cairngorm is probably the richest site in the UK for this fungus. Out of 15 samples from another study that we did, it was in 8 of them, right? So this is crazy. This thing has been found once, the fruit body, but it might be all over the place in our mountains. It just doesn't produce fruit bodies. Okay, so that's something to watch out for when you're up there. I've never seen this. I've never seen any of these guys. If anybody finds one, please give me a call. Because what's really weird is there is another one. We have a barcode for something that we can say is a Squamanita, but there's no reference. There's no reference barcode for this species, and all of these guys are in the database. So this is a species that has not been described. This is an unknown Squamanita. It's very like this guy, but it's an unknown. So if anybody finds something that looks like that, chances are you have an undescribed species that you can then name. You could call it Squamanita something else after your mum or your brother or whatever. So there's lots of things like this up there, undescribed species. Okay, this would be a great one. Again, never seen this one. So lots to see, lots to find. My favorite group has to be these guys. This is what I've been working on for the last X years. These are fungi that grow on the roots of plants, and they're beneficial to the plants to the point where the plants cannot survive without them. Okay, it's a mutually dependent association. So the fungi cannot survive without the plants, and the plants cannot survive without the fungi. They have a particular name. They're called ectomycorrhizal fungi. Ecto meaning outside, mycorrhizal meaning fungus root. And we know this group really, really well. So finding anything in this group, we usually have a very good chance of knowing what the barcode relates to. And we've had some surprises. These are the plants which they grow with, bearberry, both the common one and the alpine bearberry, and then dwarf willow. And somebody pointed out that that points to me being a dwarf birch, but I'm actually standing in a dwarf birch forest. That's on the side of Loch Mick. Anybody who can go up the left-hand side of the loch, you're actually walking through one of the best dwarf birch forests in the UK. All of these plants form particular associations with this group of mycorrhizal fungi. And because if you look at the roots of these guys, these are in fact the root systems. They're all completely covered by fungi. You can't actually see any root material here because it's totally covered by the fungus. Okay? And the fungi go out into the soil, take up nutrients, and pass some of them back to the plant in return for sugars. So it's a mutualistic association. So these are the guys that I'm particularly interested in. Some of them that we found in the Munros study are very common enough to actually have an English name. Some of you may have eaten this. If anybody wants to eat it, remove the skin on the top. It's a laxative, so not so great. But some of them are rarer. This guy here, Xerocomus ferruginous, is widespread, but not so common. But we have some other real rarities turned up on the Munro study. This guy was actually described from Finland. And again, I've never seen this. And there were 2 records of it before we did our study. And it's on several of the Munros. I'd love to see this. But one of the more spectacular ones is this big thing. Well, big is relative, I suppose. This is Amanita Gruenlandika. No guesses for where it was described from. Okay, this is new for the UK. And this is a group we know really, really well. All right, so this is great to find this because it's up there somewhere and we will track it down. So lots of surprises and lots of good records. Just a few other ones I'd like to highlight. This fungus is universal, it's global. Wherever you have ericaceous plants like the Kaluna here, whether blaeberries or crowberries, this fungus invades the cells of the roots and goes out, takes up nutrients and brings some of them back. And the photo on the left there shows one of these weird hair roots that Heather formed, and the pink color is actually the fungus inside the cell. It just completely occupies the cell of the root. Okay, so these are symbiotic fungi growing only with ericaceous plants. I just wanted to show you how common this was. It was the 7th most common fungus found in our study. Okay, so it's ubiquitous and very, very common. The final group I want to mention has this fantastic name, Archaeo Rhizomases. And we didn't know this existed until about 5 years ago, when somebody from Sweden described this whole big group of fungi. And the name means ancient root fungi, and they're very common, I have to say. We found 53 of them. They make up over 10% of all the data we have from the Munros. And the most amazing thing is, we know nothing about them. Okay? There are hundreds, if not thousands, of this group of fungi, and we know nothing about them. There's one species being described that has actually got a species name. We didn't even know they existed until a few years ago. They're absolutely ubiquitous across the planet, and we know nothing. It's amazing. Okay, that's a few of the individual fungi that we found. I want to show you a wee bit of what we've done in relation to the communities. And to do that, I want to go back to our original question in the Benan. How is the heterogeneity above ground reflected below ground? And certainly, as I showed you, that's very strongly so on Benan. But what about the rest of the Munros? So this is going to take a wee bit of explanation. This is what's called an ordination diagram. And it's a way of taking this enormous amounts of data and simplifying it. Okay, and I'll talk you through this. Each symbol on here is in fact a sample. Okay, so there's 219 dots on there, or 219 points. Each one is a sample, and that sample is a community of fungi. And what this approach does is it relates each of the samples together, so that the symbols which are closer together are more similar with respect to the communities in those samples. So for example, these two here, their communities must have been very, very similar in order for them to appear close together on the diagram. Whereas these two must be quite dissimilar. Okay, so 2 points on the diagram which are close together, similar communities, further away, distinct communities. So how does this relate to what we looked at? We looked at 3 main vegetation types. Okay, so we've got grasslands, we've got moss heath, and then we've got shrub heath. And there is some overlap, but when you look at the general pattern, the communities within each of these vegetation types must be pretty similar in order to be able to relate that across the diagram. So out of those 55 Munros, our ideas about the close associations between above and below ground seem to be upheld. Okay. The lucky dips, I was a bit disappointed because this is a diagram with outliers all over the place, but a lot of them seem to fall into our 3 main vegetation types. But fantastic set of data. So, just to summarize things a wee bit, we've got 73 participants. We've got 2,748 fungi. That's about 38 fungi per person, which is a pretty good ratio. The average number of species in a sample was 80. The most came from, I'll probably pronounce it wrong, Ben Vian. Is that close? Ben Vane. I had it written down. Okay, so 358 species came from that one mountain. So this is an amazing study. It's quite unique, actually. So it's hugely increased our knowledge about alpine fungi, not just in Scotland, but it's a unique study globally. So it's such an amazing study, this, with the help from all the volunteers and Plantlife. So where do we go from here? Well, we've got lots of data now because we've only had the data back about 3 weeks. And we've got all the soil data, which we've got back as well. So we need to look at how the soil data influences the fungal communities, because then we can look to see what drives the community structure. And this is where it becomes super important with climate change, because you can see what the drivers are of these communities. Are they susceptible to changes in rainfall, to temperature, all sorts of things. And that's one of the reasons why we're doing all this work. So once we've done all that, and then the plan is we'll go and sample all the Munros in Scotland. So that will start next year. So anybody who would like to take part in this, the Plantlife will set up another website, and our plan is to do all 281 Munros with the volunteers, which would be amazing. That's the idea. So just anyone who took part in this, anybody who was involved in any way, a huge thank you. And also thank you for listening, and thanks for the invitation.
And that is mind-boggling. I don't think I'm going to walk on the hill. I'm going to be so careful about where I tread, what I'm treading. Human impact, and you didn't. It's early. Obviously, it's early days in the study. So Ben Arn and Ben Vane, they're very remote Munros. They don't get a lot of foot traffic compared with some of the others you looked at. And is that evident in the study?
No.
No.
I mean, if you think about it, the Ben Arn study was done on 99 plots. Okay, that area is, I think it's 5 square kilometers, or 10. I think it's 10 square kilometers. So the amount of area up there is huge. There's no doubt that paths have where you've got soil compaction has a big impact. But with respect to the impact, the overall impact, it must be very low indeed.
Is that so?
What we do know has had a huge impact is nitrogen deposition. And Scotland is very lucky in that we still have large areas which are near pristine up in the mountains. We've done lots of studies from northwest Scotland, central, lowland, Lake District and Wales, and the differences between areas, shall we say, with respect to the vegetation is huge. And you can relate that directly back to the amount of nitrogen that's going on to the system. If you know Racemitrium moss, right, it forms big cushions like this over huge areas sometimes up here. We did a study where we looked at one of the factors affecting the growth and decomposition of the moss, because it's a habitat in the UK which has suffered massively. It's a very important habitat for all sorts of things, including dotterel, nests on Racemitrium heath. And dotterel has declined phenomenally. Racemitrium heath has declined phenomenally. We did a transect. We did a study from all the way down, and when we had little mesh bags of moss that we put under the moss and then measured decomposition over 2 years. When I was in Wales on Snowdon, it was hard to find patches of moss big enough to put the moss bags under. It's so degraded. It's incredible. A couple of years ago, we ran a workshop in the Institute in Aberdeen, and we had folk from all over the UK. And there was a guy there from the Lake District, and we were talking about climate impacts. And he said, "We don't have climate impacts in the Lake District." And we're like, "Okay." He said, "We have sheep, and they are the dominant force, right?" So the grassland, which I showed you, small inset, that's basically what they have. They have lost an incredible amount of heterogeneity, and it's that heterogeneity which supports the biodiversity. It's incredible what's happening.
It's mind-boggling. So this research, it's about 3 weeks old. That's what the
date. I forgot what date it is actually. It might be. I think we got the date about a month ago, maybe something like that.
So all of the path rebuilding, the hill tracks, ski development, all of the disturbance that we've been participating in over the years, we have had no idea at all about what we're moving, what we're interfering with. Yeah,
it has to be something which has a big impact. So this is why I mentioned nitrogen deposition, because you've got this weird. There's more nitrogen being deposited on the tops of the hills than at the bottom. It's this weird phenomenon that you got. So that's the other thing that we're going to try and look at, is because a lot of our data about nitrogen deposition is modeled. It's very easy to get data from some areas, but from all the mountaintops,
There's going to be a growth in Munro bagging, isn't it? Particularly when you start your next survey. Does anyone, Helen?
Yeah, I'm curious, that nitrogen deposition.
Can you?
Sorry.
That's okay.
How does it impact vegetation? Is it a fertilizing effect? Do you get more vegetative growth?
Yeah, we did some work on this. We published it a couple of years ago. The Racemitrium responds by growing faster, as you would predict, because it's very N limited up there. But the decomposition speeds up even faster. So having a moss mat like that, it grows faster, but the decomposition grows like this. So it leads to fragmentation, and then other species come in. And because there's more nitrogen in the system, you've got grasses. I mean, it's well documented. This is actually the process. But we showed that it was actually decomposition is faster. It's scary.
And the impact on Snowdon, was that due to trampling?
No, that's it. It's primarily N deposition. Yeah. It's primarily a nitrogen deposition.
Yeah. Any more questions from the audience? There's some online, but yes, please. Could you take your mask off so we could hear you? Thank you.
Right at the beginning of the talk, you mentioned how many species there were in the ground soil. So presumably when you ground up your soil samples, you must have had the DNA of hundreds and thousands of things that weren't fungi. Did you look at those at all, or were they just discarded?
Yeah, not in this study. On the Ben Ann study, we looked at everything that we could. So
a lot of protozoa.
Yeah, well, funnily enough, well, that's a very good question, because protozoa have been reclassified. I've actually just written something called spatial congruity across 7 kingdoms. So we looked at 7 different kingdoms. So we've got bacteria, we've got archaea, we've got fungi, plants, and then various weird groups. So across all the kingdoms, this connection between above and below ground holds. So we found all sorts of things, lots of things I had to look up because I've never heard of them before. So depending on which part of the DNA that you look at, it gives you access to different groups. Because with the fungi, we can just pull out the fungi DNA that we want. Yeah.
Ian.
Good evening. 3 parameters I wonder.
Could you speak up, Ian, because we're listening.
3 parameters that I wonder to what extent it's being influenced by what you're doing. Latitude, geology underneath, and moor burn. That's the kind of soak like the latter one here.
Yeah, moor burn doesn't get up there. You can have wildfires, because this is all above, it's way above the tree line here.
So if I change moor burn to wildfire.
Yeah.
So what's...
Sure. If you burn the system, it changes.
So latitude and the underlying geology.
Yeah, latitude's a good one. Scotland is brilliant because you've got such strong gradients. You've got east-west gradients of temperature and rainfall. That's a fantastic gradient you've got there. The latitude actually has more relationship to, or I should say, nitrogen deposition comes into play when you look at latitude. We haven't, within Scotland, I've had various students looking at various fungal communities, and the biggest gradients we have are the east-west gradients. Latitude, we haven't really found as a big factor yet. Certainly, if you go, I mean, we also do a lot of work on continental Europe, and then obviously latitude has a big impact because you have much longer gradients. Or it's not a gradient, but
Fascinated by the comment about picking up a species with the species named Greenland.
Yes. Yeah.
Yes, Peter?
My question is perhaps more a theological question than a scientific one, but what was there in the beginning? If you go to, say, Greenland, or where the permanent ice cap is disappearing,
Yeah.
if you sampled the land just below that, would you find the same variety of species there?
Mm-hmm. It depends. I mean, there's obviously been a lot of work there is a lot of work going on, I should say, in relation to permafrost thawing. And they're finding all sorts of things preserved in permafrost from, thousands of years ago. With respect to glacial regress, what they find mainly is you've got very sterile conditions, and there's a whole very good sequence of colonization. And this is where the fungi come into play with the mycorrhizal fungi, because a lot of the pioneer plants need mycorrhizal fungi, so you need the plant and the fungus at the same time. If you go back 12,000 years in Scotland, for example, or a bit longer, we were basically white clean. So we needed both the fungi and the plants to come back at the same time to give us what we have. We've got some very strange data to suggest that things were not white clean. And if anything is going to survive, it's these Arctic alpine plants with their associated fungi. And I have data that I can't explain, which I'd love to investigate further, which would suggest that there was refugia somewhere.
Yes, please.
I'm concerned about the effect of climate change on biodiversity. And I imagine that's along with that fungi are at the base of so many food chains for both flora and fauna. So I'm wondering what the impact is of climate change on the fungi. But since your data is only 3 weeks old, I imagine it's very much a cross-section rather than longitudinal study. Do you have any thoughts of this?
Yeah, for sure, but not in relation to this.
Or what the implications might be.
Sure. There's been some. Britain is amazing because we've got such long-term data sets, and I mean, the Victorians were brilliant for collecting all sorts of things, and it carried on not just with the Victorians. And there was a chap in England who recorded the fruit bodies, the mushrooms, in the area he was in over 60 years. And using that data set, they have managed to show that the length of the fruiting season has increased. So they're appearing earlier and later. So the whole phenology of fruiting for the fungi has changed. And that's potentially in relation to climate change. And that's been shown both in the UK and also in Scandinavia. So from that point of view, things have changed. You're thinking of other things coming in potentially.
And also things disappearing.
Yeah. Fungi are really difficult. For a fungus to be extinct, basically it hasn't to be seen in 50 years. Because the fruit bodies appear so sporadically, so randomly, and quite often, for years. The Strangler, for example, there was one fruit body in 1957 found. And people would think it was a one-off. It's just that was the one mycelium that existed, and now it's extinct. We find it all over the place. It just doesn't produce fruit bodies. So cryptic organisms like those are really difficult to work on to say when something goes extinct. So that's a tricky one.
New species coming in?
Yeah, that's another very interesting question with respect to the UK. And I personally think there's actually very little evidence that new species are appearing naturally. We certainly bring things in. We have something like 600,000 hectares of spruce in Scotland. The numbers of species which are specific on spruce of the group I work on, there's 2. Right? On the continent, there's loads. So if spore rain was coming in, there would be a lot more. So I actually think spore rain from the continent is a very rare event. So I'm not sure how fortunate we're going to be with natural colonization.
Thank you.
Very good. We've got some questions on line 4, if I can start with them. So from Greenshanks Music, thanks, great talk. We'd love to know if the soil around the very rare remnant montane willow ledges contains significantly different fungal species, and how important is it for the survival of the willow?
Great question. Willow is one of these guys that is very unusual. It forms most plants will only form one type of association with these fungi. So there are 3 main groups. I showed 2 of them tonight. You've got the ericaceous ones, you've got the ones I showed you, and then there's another group. Willow can form both those groups, which is really unusual. Only willows, poplar, and eucalyptus can do it. The rare willows in the mountain scrub is a fascinating topic, and we are actually working with people on this. Salix herbacea, the dwarf willow, has a huge diversity of these symbiotic fungi growing with it. It's got the highest diversity of any of the plants which form that particular association. So, but there's nothing we know which grows specifically on the rare willows.
Okay, right. Good question. Thanks for that. And then, well, what, if any, were the big surprises from the study, or did it mainly confirm your assumptions?
The biggest surprise was the number of samples we got. We had bets on how many we were going to get back. So I was very pleased at what we got. Funnily enough, the one mountain that we didn't get was Loch Nagar, which is one of our closest ones to Aberdeen. And a couple of colleagues tried twice and got beaten back by the weather, unfortunately. So that was one of the biggest surprises. With respect to the fungi, it's always the species that we can identify which are the biggest surprises. The Amanita Gruenlandika, that was a great surprise. Yeah.
Right.
Yeah.
Okay. And then there are a couple here which I think you'll be particularly interested in. So, where will a link appear for the next volunteering to be part of the study? So,
on Plantlife, basically, I think.
Yeah. And what will be the length of window for collecting Munros samples in 2023?
Yeah. This year or last year, it was from June to September. Okay. We haven't included the late snow beds for various reasons. So June to September.
Okay. Yeah. Right. Peter. Yeah.
I'm a bit puzzled about the barcodes.
Yeah.
Are they internationally recognized?
Yes.
Okay.
Yeah. They're the barcode which we use for fungi is internationally recognized as the barcode for fungi. Yeah. And one reason why we know so much about the group I work on is in 2001, maybe, we set up a group to collect all the barcodes from that particular group of fungi in Europe. And we set ourselves the goal that within 5 years, we'd have 95% of them. And we cheated a wee bit because we collected some of them in Southern Europe. It's the same species, but we got the barcodes from collections that we had made in Southern Europe. So that's quite unique. That database was a curated database of authentic material, and every barcode was linked to a specimen in a herbarium. But it's expanded since then. So, but yeah.
Helen, could you, Matt, please?
Oh, sorry.
So we can hear you.
Is it full genetic sequencing that you're doing, or is it SNPs?
It's a tiny little piece of the DNA. It's just the ITS, if you know what that is. Yeah. So it might only be 600 base pairs. So it's a tiny little piece, but it's unique, fortunately.
Yes, please. Yeah.
So these plants that have their fungi growing on the root tips, does that mean if a seed lands in a bit of soil that doesn't have the right kind of
Exactly.
It won't germinate?
It won't germinate. And that's one of the problems that we have with woodland expansion, because we're taking seedlings or seedlings from nurseries, which are effectively sterile, and putting ectomycorrhizal plants into heather moorland, and there's no fungi there.
Doesn't have the right fungi.
Yeah. So, and we're really surprised that they die.
So the fungi have to be there first.
No, the plants from nurseries are usually stuffed full of nutrients. So they'll sit there for a couple of years and they'll internally recycle. And then after a couple of years, they'll die. But if they're lucky and spores come in within that period of time, or they get contacted somehow, then they'll be okay. Yeah.
So you need donations of soil from the.
Then you're tricky because you're introducing all sorts of other things.
Yeah.
Yeah. I'm working with the Woodland Trust at the moment on Ben Shield egg, and it's a really they're trying to plant up this hillside that is just, it's been so degraded. But we found out there's Salix repens, the creeping willow, all over the place, and it can support the right fungi. So we're going to use those as little sites of inoculum, basically. So you can plant pine and birch around them. Yeah. So they're very lucky.
That really interests me because I do a lot of volunteering at the tree nursery at Forest Lodge.
Okay.
With Cairngorm's Connect and the RSPB. So we grow, we've got hundreds of thousands of little aspen, birch, and willow species for helping to recolonise the montane habitat and that. So are we wasting our time?
That's a dangerous question.
Don't answer it, please.
No. You've got the willows and the poplar. The willows and the poplar have the advantage that they can pick up one of the most common associations, and then they can then be colonized by the ectomycorrhizal fungi. So willows and poplar, not a problem. The birch need aspen, poplar, yeah. Mm-hmm. So it can pick up the more common one. So as long as you've got other plants there which are not ericaceous, then they will have the fungi which can colonize them, hopefully.
Yeah. Last question, please, from you.
Sorry. A friend of mine running a nursery specific to native species, particularly Scots pine. He developed a technique where he would go into a pine forest and collect material and then introduce it to the irrigation system, and the survival rate was just phenomenal.
Okay.
The plants are coming out. I take the point about might be taking other things with it, but at least his survival rate, which relates to the amount of grant that might or might not have to be paid. So just thought it, if people want, they can go and
Collect what though?
He was collecting humus around at the base of Scots pine, native pine, just enough to multiply through the irrigation system. And he's been able to get this into the nursery on a regular basis, all the time. And his survival rates are phenomenal.
I'm just wondering how he got it into the irrigation system. That would work for sure.
Well, you're soaking it in bags. He's filling that material into an open weave bag.
Okay.
And dumping it in the water. But the person you need to speak to is the guy. His name is Rodney Shearer.
Okay.
And his name has been given to Colin.
Okay.
I can give you more information later.
Sure. Thank you for that. Andy, that has been fascinating, mind-boggling. I think the Hutton Institute and the esteem it's held in this community has just risen through what you've explained to us in the last hour. As I say, I don't think I'll tread on the hill again with quite the same level of comfort that we're knowing what's below. Andy come over from Aberdeen for the evening. I'm afraid there's very little we can, but please accept that on behalf of the whole community.
Super.
And thank you so much for
Many thanks.
for the talk. Thank you so much.
Thank you.
Andy's chauffeur, who drove him over this evening, is a good friend, and it's Fiona Davison, daughter of John Davison, who is a good friend of Andy. So Fiona is coming back home in many ways, back to Boat of Garten. So your talk was to be the last, Andy, of the winter series, but we do have one more, and that's in 2 weeks' time, on the 21st of March, and it's, should we call it the second part of Peter Mackay's talk? Peter, you had a spellbound for about 20 minutes. We can't wait for you to come back and finish the talk. So that's not to be missed in 2 weeks' time. And finally, I want to thank all of the talks team, for Graham, for managing the IT, for Alan, who has had to disappear off, and for the ladies who were helping at the door, but particularly to Mike Martin, who is in his bed right now listening to this, and he should be stood here. So Mike, we hope you're better very, very soon and you get away. Thank you all for coming. It's been lovely to have you with us. Thank you so much.
Good.
So much.